Hello and welcome, dear students. Today we are going to discuss the topic functions of pituitary hormones. The main objectives of today's lecture are to get a brief idea about the pituitary gland, to discuss about various families of pituitary hormones, to discuss about different hormones of the pituitary gland, and finally we will describe in detail about functional understanding of pituitary hormones. To begin with, let us first discuss about pituitary gland hormones. The pituitary gland, popularly known as master gland till recently, is a pea-sized gland that regulates various physiological functions in humans by secreting various hormones. The anterior pituitary regulates several physiological processes including stress, growth, reproduction and lactation. Its regulatory functions are achieved through the secretion of various peptide hormones that act on target organs including the adrenal gland, liver, bone, thyroid gland and gonads. The anterior lobe of pituitary secretes 6 principal hormones, namely follicle stimulating hormone or FSH, luteinizing hormone or LH, adrenocorticotropic hormone or ACTH, thyroid stimulating hormone or TSH, prolactin and growth hormone, while as posterior lobe secretes antidiuretic hormone or ADH and oxytocin. Now, the important families of pituitary hormones. The pituitary hormones are divided into 4 different families like glycoproteins, peptides, pro-opiomelanocortin and non-peptidic hormones. The function of all the hormones will be discussed as per the family. Glycoprotein family. Glycoprotein hormones are among the largest hormones known to date. They include TSH, FSH, LH and human chorionic gonadotropin or HCG produced by the placenta. These hormones are heterodimeric glycoproteins consisting of a common alpha subunit and a unique beta subunit which confers the biological specificity of each hormone. Thyroid stimulating hormone or TSH is a glycoprotein synthesized and secreted from thyrotropes of the anterior pituitary gland. Thyrotropes synthesize and release TSH in response to thyrotropin releasing hormone or TRH stimulation. TRH is synthesized in the paraventricular nuclei of the hypothalamus predominantly by parvicellular neurons and is released from nerve terminals in the median eminence. TRH binds to a GQ11 protein coupled receptor which activates phospholipase C, leading to increased phosphoinositide turnover, calcium mobilization and release of thyroid stimulating hormone into the circulation. TSH binds to a G protein coupled receptor in the thyroid gland, activating adenylate cyclase and leading to increased intracellular cyclic 3,5 adenosine monophosphate or CAMP formation and stimulation of the protein kinase A. signaling pathway. TSH stimulates all the events involved in thyroid hormone synthesis and release. In addition, it acts as a growth and survival factor for the thyroid gland. The release of TSH from the anterior pituitary gland is under negative feedback inhibition by thyroid hormone, particularly triiodothyronine. Gonadotropins, that is follicle stimulating hormone and luteinizing hormone. The gonadotropic hormones LH and FSH are synthesized and secreted by gonadotropes of the anterior pituitary in response to stimulation by gonadotropin releasing hormone, that is GnRH. Most of the gonadotropes produce both, that is LH and FSH, with a fraction of the gonadotrope population producing LH or FSH exclusively. GnRH is synthesized and secreted by the hypothalamus in a pulsatile manner. GnRH binds to the GnRH GQ11 protein coupled receptor on pituitary gonadotropes and produces activation of phospholipase C, leading to phosphoinositide turnover and calcium ion mobilization and influx. This signaling cascade increases transcription of FSH and LH alpha subunit and beta subunit genes and increases the release of FSH and LH into the circulation. FSH and LH exert their physiological effects on the testes and ovaries by binding to G protein coupled receptors and activating adenylate cyclase. Among the target cells for gonadotropins are ovarian granulosa cells, theca interna cells, testicular Sertoli cells, and Leydig cells. The physiological responses produced by the gonadotropins include stimulation of sex hormone synthesis, that is steroidogenesis, spermatogenesis, folliculogenesis, and ovulation, that is the release of the egg. Therefore, their central role is the control of reproductive function in both males and females. GnRH controls the synthesis and secretion of both FSH and LH by the pituitary gonadotrope cell. Gonadotropin synthesis and release as well as differential expression is under both positive and negative feedback control by gonadal steroids and gonadal peptides. Gonadal hormones can decrease gonadotropin release both by decreasing GnRH release from the hypothalamus and by affecting the ability of GnRH to stimulate gonadotropin secretion from the pituitary itself. Estradiol enhances LH and inhibits FSH release, whereas inhibins A and B, gonadal glycoprotein hormones, reduce FSH secretion. The complexity of the regulation of synthesis and release of anterior pituitary hormones is best illustrated by cyclic nature of FSH and LH release. During the luteal to follicular phase transition, pulses of GnRH release occur every 90 to 120 minutes and FSH secretion predominates. In the mid to late follicular phase, GnRH frequency increases to 1 pulse every 60 minutes, favoring LH secretion over FSH. After ovulation, ovarian progesterone production predominates. Progesterone increases hypothalamic opioid activity and slows GnRH pulse secretion. This slower GnRH pulse pattern, with 1 pulse per 3 to 5 hours, favors FSH production. However, at the same time, estradiol and inhibin A produced by the corpus luteum inhibits FSH release, leading to increased FSH stores. With involution of the corpus luteum and the sharp decline in estradiol, inhibin A and progesterone, the frequency of GnRH pulse secretion is increased. In the absence of estradiol and inhibin A, inhibitors of FSH release, a selective FSH release predominates and initiates the next wave of follicular development. Now, the peptide family. Peptide hormones or protein hormones are hormones whose molecules are peptides or proteins, respectively. The latter have longer amino acid chain lengths than the former. These hormones have an effect on the endocrine system of animals, including humans. Most hormones can be classified as either amino acid based hormones, that is amine, peptide, or protein, or steroid hormones. The former are water soluble and act on the surface of target cells via second messengers. The latter, being lipid soluble, move through the plasma membrane of target cells, both cytoplasmic and nuclear, to act within their nuclei. All the peptide hormones are synthesized as prohormones and often contain superfluous amino acid residues that were needed to direct folding of the hormone molecule into its active conformation or configuration, but have no function once the hormone folds. Specific endopeptidases in the cells cleave the prohormone just before it is released into the bloodstream, generating the mature hormone or the active hormone form of the molecule. Mature peptide hormone then travel through the bloodstream to all the cells of the body, whereas they interact with specific receptor on the surface of their target cells. Dear students, now the growth hormone. Growth hormone is a 191 amino acid peptide hormone with a molecular weight of approximately 22 kilodaltons and structural similarity to prolactin and chorionic somatomamotrophin, a placental derived hormone. Growth hormone exists in various molecular isoforms, and this heterogeneity is reflected in the wide variability in GH levels determined by different immunoassays. However, it is the 22 kilodalton that is the principal form with physiologic effects found in humans. Growth hormone is released from the somatotrophs, an abundant cell type in the anterior pituitary gland. Growth hormone is released in pulsatile bursts, with the majority of secretion occurring nocturnally in association with slow wave sleep. The basis of the pulsatile release of growth hormone and the functions of this pattern are not fully understood yet. However, nutritional, metabolic, and age-related sex steroid mechanisms, adrenal glucocorticoids, thyroid hormones, and renal and hepatic functions are all thought to contribute to the pulsatile release of growth hormone and appear to be essential in achieving optimal biological potency of the hormone. Most of the growth hormone in the circulation is bound to growth hormone binding protein. The physiological effects of growth hormone. Growth hormone can have direct effects on cellular responses by binding to the growth hormone receptor at target tissues, and indirectly by stimulating the production and release of IGF-1, a mediator of several of growth hormones effects at target tissues. IGF-1 is a small peptide about 7.5 kilodaltons structurally related to proinsulin that mediates several of the anabolic and mitogenic effects of growth hormone in peripheral tissues. The most important physiological effect of growth hormone is stimulation of postnatal longitudinal growth. Growth hormone also plays a role in regulation of substrate metabolism of adipocyte differentiation, maintenance and development of the immune system, and regulation of brain and cardiac functions. Now the prolactin hormone. Prolactin hormone, or PRL, is a polypeptide hormone synthesized and secreted by lactotrophs in the anterior pituitary gland. The lactotrophs account for approximately 15 to 20% of the cell population of the anterior pituitary gland. However, this percentage increases dramatically in response to elevated estrogen levels, particularly during pregnancy. Prolactin levels are higher in females than in males, and the role of prolactin in male physiology is not completely understood. Plasma concentrations of prolactin are highest during sleep and lowest during the waking hours in humans. Now, the physiological effects of prolactin hormone. The physiological effects of prolactin hormone are mediated by the prolactin receptor, a single membrane bound protein that belongs to class 1 of the cytokine receptor superfamily. Prolactin receptors are found in the mammary gland and the ovary, two of the best characterized sites of prolactin actions in mammals, as well as in various regions of the brain. Activation of the prolactin receptor involves ligand-induced sequential receptor dimerization. Prolactin-mediated activation of prolactin receptor results in tyrosine phosphorylation of numerous cellular proteins, including the receptor itself. The main physiological effects of prolactin are stimulation of growth and development of the mammary glands in females, synthesis of milk, and maintenance of milk secretion. Prolactin stimulates glucose and amino acid uptake and synthesis of milk proteins, beta casein and alpha lactalbumin. The milk sugar, lactose, and milk fats by the mammary epithelial cells. During pregnancy, prolactin prepares the breast for lactation. The production and secretion of milk is prevented during pregnancy by the high progesterone levels. Additional effects of prolactin include inhibition of GnRH release, progesterone biosynthesis, and luteal cell hypertrophy during pregnancy. Prolactin also modulates reproductive and parental behavior. Now, pro-opiomelanocortin, or POMC derived hormone family. POMC is a precursor prohormone produced by the corticotrophs of the anterior pituitary gland. The production and secretion of POMC derived hormones from the anterior pituitary are regulated predominantly by corticotropin releasing hormone, or CRH, produced in the hypothalamus and released in the median eminence. CRH binds to a G protein coupled receptor whose actions are mediated through activation of adenylate cyclase and elevation of CAMP production inside the cell. Two types of remarkably homologous, that is approximately 70% of amino acid identity, the CRH receptors have been identified. Both CRH1 and CRH2 receptors belong to the family of transmembrane receptors that signal by coupling to G proteins and use CAMP as a second messenger. Stimulation of POMC synthesis and peptide release is mediated by CRH1 receptor, which is expressed in many areas of the brain as well as in the pituitary gonads and skin. CRH2 receptors are expressed on brain neurons located in neocortical, limbic, and brainstem regions of the central nervous system, and on pituitary corticotropes and in peripheral tissues, like in examples are cardiac myocytes, gastrointestinal tract, lung, ovary, and skeletal muscles. The role of CRH2 receptors is not completely understood yet. POMC is post-translationally cleaved to ACTH, beta endorphin, and endogenous opioid peptide and alpha, beta, and gamma melanocyte stimulating hormone, or MSH. The biological effects of POMC derived peptides are largely mediated through melanocortin receptors, that is MCRs, of which 5 have been described. MC1R, MC2R, and MC5R have defined roles in the skin, adrenal steroid hormone production, and thermoregulation, respectively. MC4R is expressed in the brain and has been implicated in feeding behavior and appetite regulation. The roles of MC3R is not well defined till date. Now the adrenocorticotropic hormone, or ACTH. The main hormone of interest produced by the cleavage of POMC is ACTH. The release of ACTH is stimulated by psychological and physical stress such as infection, hypoglycemia, surgery, and trauma, and is considered critical in mediating the stress or the adaptive response of the individual to stress. ACTH is released in small amounts and circulating levels average 2 to 19 picomoles per liter in healthy individuals. The hormone is released in pulses with the highest concentrations occurring at approximately 4 a.m. and the lowest concentrations in the afternoon. ACTH released into the systemic circulation binds to G protein coupled receptors, part of the MCR superfamily, that is MC2R, and activates adenylate cyclase, increases CAMP formation, and activates protein kinase A. The physiologic effects of ACTH at the adrenal cortex are to stimulate the production and release of glucocorticoids like cortisol, and to a lesser extent mineral corticoids like aldosterone. Although all 5 MCRs can bind ACTH to some extent, MC2R binds ACTH with the highest affinity and is expressed almost exclusively in the adrenal cortex. Thus, it is considered the physiologic ACTH receptor. The release of cortisol follows the same diurnal rhythms as that of ACTH. The feedback inhibition of ACTH and of CRH released by cortisol is mediated by glucocorticoid receptor binding in the hypothalamus and in the anterior pituitary gland. Dear students, let's now discuss the melanocyte stimulating hormone, or MSH. Alpha MSH, or alpha melanocyte stimulating hormone, is produced by the proteolytic cleavage of POMC, mainly in the pars intermedia of the pituitary gland. Only small amounts of alpha MSH are produced in the pituitary under normal conditions. Melanocortin peptides exert their effects through MC1R found in melanocytes, which are key components of the skin's pigmentary system, endothelial cells, monocytes, and keratinocytes. Binding of alpha MSH to MC1R activates adenylate cyclase, which in turn causes an increase in intracellular CAMP. This is the classic pathway by which alpha MSH is believed to increase melanin synthesis in melanocytes and thereby imparting the black color. The peripheral production of alpha MSH by non-endocrine cells, particularly by melanocytes, has been described. The involvement of this paracrine system in the development of skin cancer has received considerable attention because of the localized production and paracrine actions of this peptide and the greater expression of MC1R in melanoma than in normal skin. Now, the beta endorphin. Beta endorphin, the most abundant endogenous opioid peptide, is another product of POMC processing in the pituitary. The physiologic effects of this opioid peptide are mediated by binding to opiate receptors. Because these receptors are expressed in multiple cell types in the brain as well as in peripheral tissues, their effects are pleiotropic. The physiologic actions of endorphins include analgesia, behavioral effects, and neuromodulatory functions. Among the effects of endocrine functions is inhibition of GnRH release. Endogenous opioids have also been implicated in the mechanisms involved in alcohol and drug addiction and have led to therapies such as the use of naltrexone, an opiate receptor antagonist, in the management of alcohol dependency. Now, the non-peptidic hormone family. The posterior lobe does not synthesize hormones, but it contains a large number of unmyelinated nerve fibers. These fibers form the hypothalamo-hypophyseal tract within the pituitary stalk. These neurons synthesize predominantly two that are 9 amino acid long, vasopressin and oxytocin. These two hormones are then transported as protein bound secretory granules, neurophycins, down the nerve fibers to accumulate at the nerve terminals within the posterior lobe. Now, the arginine vasopressin. Arginine vasopressin, or AVP, also known as antidiuretic hormone, is the other neuropeptide produced by magnocellular neurons of the hypothalamus and released from the posterior pituitary. The principal effect of AVP is to increase water reabsorption by enhancing permeability to water in the distal convoluted tubules and the medullary collecting ducts in the kidney. In addition, AVP increases vascular resistance. This function of AVP may be important during periods of severe lack of responsiveness to other vasoconstrictors, as may occur during severe blood loss, that is hemorrhagic shock, or systemic infection, that is sepsis. The circulating concentrations of AVP range from 1.6 to 6 nanograms per liter. Now, the physiological effects of arginine vasopressin. The main target site of ADH is the collecting duct in the kidney. Water permeability in the collecting duct is relatively low compared with that in the proximal tubule and the thin descending limb of Henle's loop. In the proximal tubule and the descending limb of Henle's loop, The water channel protein aquaporin 1 is constitutively expressed both in the apical, that is luminal, and basolateral, that is interstitial membranes of epithelial cells. The proximal tubule is responsible for resorbing approximately 90% of filtered water. Resorption of the remaining 10% of filtered water in the distal collecting ducts is under tight control by AVP. Although it may seem that only a small fraction of the total filtered water reabsorption is under AVP control, water permeability of the collecting duct can be dramatically increased within a few minutes through the production of CAMP following AVP binding to V2 receptors in the basolateral membrane of the principal cells. The importance of AVP is better understood in terms of the total amount of urine that would be excreted in its absence. For example, in a healthy individual, an average of 180 liters of glomerular filtrate is formed per day. Thus, without AVP mediated resorption of 10% of filtered water in the distal collecting ducts, urine output would be close to 18 liters per day. This is tenfold higher than the volume of urine output, which is usually 1.2 to 2 liters per day under normal conditions. The increase in CAMP that is stimulated by AVP binding to the receptor located in basolateral membrane activates protein kinase A and subsequently the phosphorylation of AQP2, another protein. Phosphorylation of AQP2 is essential for its movement from cytoplasmic pools and its insertion in the luminal, that is apical epithelial cell membrane of the collecting duct cells. The result is an increase in the number of functional water channels in the luminal membrane, making it more permeable to water. Thus, AVP mediated insertion of AQP2 into the luminal membrane results in water conservation and urine concentration. This event is a short-term regulation of water permeability in response to an increase in circulating levels of AVP. In addition, AVP is thought to regulate water permeability over hours to days as a result of an increase in the total cellular amount of AQP2 caused by increased protein synthesis. AQP2, one of several members of the aquaporin family, is exclusively expressed in the collecting ducts of the kidney. It is the only aquaporin that is directly regulated by ADH via the V2-AVP receptor. Water that diffuses into the cells through AQP2 exits through the basolateral side through aquaporin 3 and aquaporin 4, eventually entering the vasculature. AQP3 and AQP4 are constitutively expressed in the basolateral membranes of the collecting ducts. Now, the water reabsorption through this mechanism is driven by the hydroosmotic gradient generated by a countercurrent mechanism in the renal medulla. The result is an increase in the concentration and reduction of urine volume, which minimizes urinary water loss. This antidiuretic mechanism may increase urine osmolarity to approximately 1200 millimoles per liter and reduce urine flow to approximately 0.5 ml per minute. Without AVP mediated effects, the principal cells of the collecting duct are impermeable to water, resulting in large volumes of diluted urine entering the collecting tubules from the ascending limb of the loop of Henle. As mentioned earlier, this could translate into excessive urine output and reduced urine osmolarity. AVP also binds the V1 receptor expressed in vascular smooth muscle, producing contraction and increasing peripheral vascular resistance. The hormone is known as vasopressin because of these vasoconstrictor effects. In particular, renal medullary blood flow has been demonstrated to be under AVP regulation. AVP circulates unbound and is distributed in a volume approximately equal to that of the extracellular spaces. Because of its relatively low molecular weight, AVP penetrates peripheral and glomerular capillaries readily, so the urinary excretion rate of AVP is extraordinarily high. Now, dear students, let us discuss the physiological actions of oxytocin hormone. The two main target organs for oxytocin's physiological effects are the lactating breast and the uterus during pregnancy. In the lactating breast, oxytocin stimulates milk ejection by producing contraction of the myoepithelial cells that line the alveoli and ducts in the mammary gland. In the pregnant uterus, oxytocin produces rhythmic smooth muscle contractions to help induce labor and to promote regression of the uterus following delivery. Oxytocin analogs may be used clinically during labor and delivery to promote uterine contractions and during the postpartum period to help decrease bleeding and return the uterus to its normal size, that is, uterine involution. The physiologic effects of oxytocin in the pregnant uterus are augmented by a dramatic increase in sensitivity to the hormone during the onset of labor. This increased sensitivity to oxytocin is caused by an increased density, that is, upregulation of oxytocin receptors in the uterine muscles. Receptors levels can be 200 times greater at the onset of labor than in the non-pregnant uterus. Because of this increase, levels of oxytocin that would normally not be effective can produce forceful uterine contractions towards the end of pregnancy. This increased density of oxytocin receptors is mediated by steroid hormone regulation of oxytocin receptor synthesis. Responsiveness of the uterus is also enhanced by increased gap junction formation between smooth muscle cells, facilitating conduction of action potentials between one cell and the next, and by increased synthesis of prostaglandin, a known stimulator of uterine contraction towards the end of gestation. All of these factors enhance myometrial contractile activity in response to oxytocin at term. Additional secondary effects that have been attributed to oxytocin are potentiation of the release of ACTH by CRH, interaction with the AVP receptor to produce vasoconstriction, stimulation of prolactin release, and an influence on maternal behavior and amnesia. Now, dear students, let us discuss the control of oxytocin release. The principal stimulus for oxytocin release is mechanical stimulation of the uterine cervix by the fetus near the end of gestation. Oxytocin release is also stimulated by the forceful contractions of the uterus during the fetal expulsion reflexes. Hence, the contractile activity of the uterus acts through positive feedback mechanism during parturition to stimulate oxytocin neurons, and this further increases the secretion of oxytocin. Before the oxytocin neurons can secrete oxytocin, they must be released from inhibition by other neurons containing endogenous opioids, nitric oxide, and gamma aminobutyric acid or GABA. This modulation of oxytocin release is partly caused by the declining blood levels of progesterone and increasing levels of estrogen during late pregnancy. The neurotransmitters involved in stimulating oxytocin release are thought to be acetylcholine and dopamine. Oxytocin release is also triggered by stimulation of tactile receptors in the nipples of the lactating breast by suckling. Breastfeeding generates sensory impulses that are transmitted to the spinal cord and then to the oxytocin producing neurons in the hypothalamus. The information transmitted by these sensory afferents produces intermittent synchronized burst firing of oxytocin neurons, resulting in pulsatile release of oxytocin and increase in blood oxytocin concentrations. In addition to its secretion from neurohypophyseal terminals in the posterior pituitary, oxytocin is released within the hypothalamic, supraoptic, and paraventricular nuclei. The functions of this intra-hypothalamic release of oxytocin is to control the activity of oxytocin neurons in an autocrine fashion by a positive feedback mechanism, increasing the neurohypophyseal release of oxytocin. The release of oxytocin is inhibited by severe pain, increased body temperature, and loud noise. However, environmental, hormonal, and neural mechanisms of hypothalamic hormone regulation are in play to regulate oxytocin release at the appropriate time of gestation and in response to the relevant stimuli. The role of oxytocin in males is not clear, although recent studies have suggested that it may participate in ejaculation. Dear students, it was all about today's lecture. Hope you have understood it well. Thank you. Goodbye.